Have you ever noticed a typo in a book and thought, how did that get past the editor? Well, our bodies have their own editors for our DNA, and when they make a mistake, the consequences can be much more serious. Today, we're diving into one of the most important DNA repair mechanisms, homologous recombination. Think of your DNA as two identical copies of a massive instruction manual. If one page gets torn or damaged, your cells can use the other identical copy as a perfect template to fix it. This process is called homologous recombination, or HR for short. It's an incredibly precise system that finds the undamaged copy and uses it to restore the broken DNA sequence, ensuring the genetic information stays accurate. It's like having a backup file for your most important document. But what happens when this backup system itself is broken? That's where cancer comes into the picture. Certain genes like BRCA1 and BRCA2 are the master supervisors of this HR process. You've probably heard of them in relation to breast and ovarian cancer. When these genes have mutations, the HR repair pathway becomes faulty. It's like the editor losing their glasses. They can't effectively proofread and correct errors anymore. Without a functioning HR system, DNA damage starts to pile up. The cell might try to use other, more error-prone repair methods, which is like trying to patch a torn page with tape and a random sentence. These sloppy repairs introduce even more mutations. Over time, this genomic instability can cause cells to lose control, divide uncontrollably, and eventually form a tumor. This is why mutations in HR genes are a major driver for many types of cancer. But here's the fascinating part. We can actually use this weakness against cancer. Scientists have developed drugs called PARP inhibitors. These drugs block a different, less critical DNA repair pathway. In a normal cell, blocking this backup pathway isn't a big deal because homologous recombination can still handle any major DNA damage. However, in cancer cells that already have a broken HR system, taking away their last remaining repair option is catastrophic. It's a concept called synthetic lethality. By knocking out both repair systems, we can specifically target and destroy cancer cells while leaving healthy cells relatively unharmed. This has been a game-changer in treating cancers with BRCA mutations. So, homologous recombination is this elegant, essential process that safeguards our genetic code. When it fails, it can lead to cancer, but by understanding its failures, we've opened up powerful new avenues for treatment. It's a perfect example of how studying the fundamental biology of our cells can lead to incredible medical breakthroughs. Thanks so much for watching. If you found this interesting, don't forget to like this video and subscribe for more deep dives into the science of our bodies. See you next time.